There's still some people pouring in, so I'm just letting them pour in, giving them one more minute. And welcome. Hi, my name is Michele Cosi, and just to get things out of the way, today and for probably most of the upcoming sessions, we're going to be using Cybers, which we have a recording for how to get to Cybers and stuff in the first. Cool, fantastic. And so therefore, if you did look at the requirements, is gotta have an account, and we can get things started with that. So I also do ask for people to bring their computers. It just makes things a bit easier for us to share commands here and there. Don't worry though, you can always follow along without a computer. Make this a little bit taller. I can't. Alrighty. I also did forget my nice shirt today, so you have to see me with my sweaty clothes. Okay. So, first things started. Welcome back, and also welcome to the second session of Exploring Tools for Data Analysis and AI Application in Biosciences and Genomics. To get to the wiki head, do head to github.com UA-bio data lab /bioinformatics. This is and /wiki, excuse me. This is gonna get to where all of our information is. Oh, there is a question in the chat. You might want to answer that, Jeff. This is for the previous session. So sorry, Mike. Jeff is going to get back to you in just a second. Regardless, I shared a link in the chat on all of the workshop program from well, the previous week and onwards. And if you look at the week for 9-10, we're going to talk about CLI tools for DNA and RNA sequences. Now, please do click the link here on the right, and this is going to open today's, well, reference material for today's lesson, today's workshop, excuse me, can't call it a lesson right now. Scrolling down, here's the schedule. For the next minute, I'll talk about what else, housekeeping, I have to talk about today. And then I'm going to give you a less than 5 minutes rundown of what the command line is. And we're going to start talking about quality control tools, sequence alignment tools right after that, and some honorable mentions. And I do truly need to emphasize that today is going to be a lot. I hope not to lose you halfway. Let me know when I'm going too fast. Let me know when I'm going too slow. This is this is not a one-way thing. This is going both ways. Do ask questions, and I hope to address anything and everything that you guys might want to share. So I did also read your instant feedback. Important things. A lot of you seem to be interested in transcriptomics and working with transcript data and differential expression analysis. All of this stuff is coming. It's coming down the line within this workshop series, but also because there is many of you asking for it, we're bringing in some of the data science fellows to present on these specific topics. The data science fellows are a group of people which have been together with the Data Science Institute for a long time and are currently, well, experts in their own fields, whether that is transcriptomics or law. And because there is so many of you asking for it, keep an eye out. Sometimes in perhaps November, we're going to have a couple of really in-depth workshops just for transcriptomics and DEGs, differential expression analysis, DA. I'm going to be announcing this when we have things set up for it. Okay. With that said, we do require access to Cybers today. And with excuse me, there is a message. Let me check that out. Okay. And with that out of the way, let's talk about it. Let's talk about a number of tools that I think every bioinformatician needs to know. But before we do talk about the tools, I need to talk about the data that we are using. Who here has heard of NCBI? Who here has used NCBI? Okay, all right. No, okay. I take a little head nod. What is NCBI and what did you, quote unquote, use it for?
It's a database where you can search for, I think you can search for like proteins and genes and stuff like that.
Correct. It's literally that. It's a database where there is a plethora of genes and DNA related information. Now, it is important for you to know and oh, what the hell is this? Okay, NCBI. Type in NCBI, it's just going to bring you right here. I'm going to show this real quick because NCBI has a set of search. It's a really powerful tool, but it's a set of it's a search bar that allows you to search for a number of information going from transcript information to fully constructed genomes to genes. And it's important to know how to navigate NCBI, but I'm not going to go into that today. It's something that I think bioinformaticians should know how to navigate. Now, the reason why I'm bringing this up is because the data that we are going to be using today, tomorrow, or today, the next time, and the time after is all taken by NCBI. It's in this little admonition over here. Because we have time constraints, we can't run analysis on large genomes, plants, humans, and therefore we are going to stick with E. coli, just making sure people know what E. coli is. Cool, fantastic. And I've downloaded and made available, I'm going to show you in a minute how and where, the E. coli genome, its reference annotations, specific raw reads, and some genes that you can blast against. Any of the things I've said until now sound like something you've never heard before? Let me know. Yes.
I don't know what you're talking.
Okay, fantastic. This is Jeff. He is, excuse me a second.
Not a bioinformatician.
Not a bioinformatician. And it does not surprise me that he doesn't know what any of these are. But when we're talking about genomes and related stuff, well, when you download a genome, Jeff, you have a DNA. It's essentially the entire DNA of a specific organism. Now, with the genome, you have a set of annotations. These annotations talk about the various genetic features that this organism has. Genetic features such as genes. Each genes can be made of introns, exons. I've also downloaded and processed and made available some raw reads. Now, when you do run a specific experiment, it is likely that you're going to be sequencing this organism or a set of organisms that you have collected, and you send them for sequencing. Now, the sequence is usually done by Illumina machines or PacBio machines. Illumina yields short reads, PacBio yields longer reads, and these are, and reads are small, little groups, lengths of bases, nucleotides, ATGC, ATGC. And altogether, when you build them and assemble them and pile them up, they create something called scaffolds. And then later on, you bring them all together and then you can go ahead and create genomes, but you can use reads to also do transcriptomic analysis. So thank you, Jeff, for the question. Essentially, what I wanted to say is I've taken all of the data necessary for doing some FOX assembly, FAUX assembly, and some FAUX transcriptomic analysis later down the line. Everything should be available. Now, before I show you where, how, and when, the command line in under 5 minutes. What do you see here? Alrighty, what do we see here? When you open up a command line and want to run a command, you first see your user at host machine, separated by a colon and your current location. I'm not going to get into too much detail right now. This was part of the pre-requirements. This is something that we call the shell prompt. After the shell prompt, we have the command itself, where the first thing that comes up is the command. I'm going to run a blast search here. Everything that comes after the initial command is either a flag option, an input file, or an argument. If it's a flag or an option, it's usually it usually starts with a dash, and this is an option that comes from the command itself. Knowing what the options are, you can follow it up by what the options require. In this specific case, an input file, followed by other potential flags that might be required. In this specific case, -db, which stands for database, is a nucleotide argument, and I want you to output things in an output.txt file. All of this with an e-value of 1e-5. Who didn't know how to run a command? Cool. Now, this is the literal 2-minute syntax of how to run a command. You have a shell prompt, which you largely ignore for the specific case. Then you ask for the command, and then each command comes with its flags and options. I've added a small little note here at the bottom, which tries to explain a little bit of what's going on. And essentially, it's what I have mentioned. This is a note for everybody. Spaces in paths are bad. Just don't put spaces in your paths. Don't put spaces in the names of your data as well. So now that I have rushed through it, I am going to show you where things are. Please go to de.scivers.org. Paste it in the chat as well, where you should please log in. You can follow along or not. That's completely fine. Now, once you are in Scivers, there is a tool that I have created, which has all of the applications that we're going to be using. How do you find this little app? You go to the search bar and type in Jupyter Lab Biosciences, or Bioscience, excuse me. You can see that in the app here, there is a 1, so click on that. And this is what I want you to launch. It's called Jupyter Lab Bioscience, and this is something that I curate. Click on that specific app, and over here with analysis number, give it a good name, 09, and today's also 09. You have to move a couple of windows around. Click next, where you can select parameters. Today, we're not going to be doing a lot, so I'm gonna click on 8 maximum CPU cores, 8 minimum CPU cores, and 8 minimum gigabytes of memory. We won't need a lot today. Click next over here and launch.
Did you guys be able to find the app? I was not, but don't worry about it.
Okay. It says here that the analysis has been submitted. Should be running at some point very, very soon. I'm going to click on Go to Analysis. And it's launching me into the app itself. Fantastic. We are here. Here, not here. You're not here. I know you're not. You're good? Okay. And I get thumbs up. Where were the resource parameters? 8, 8, and 8, Eric. For space, don't worry about it. 8 min cores, 8 max cores, and 8 RAM. Yep, no worries. Okay, so this is base, this is home, as I mentioned last time around. I want to now show you where the data is. Go to over here on the left panel where it says data, click on that. Says iPlant, click on that. Says home, click on that. And then here it says your username or shared. Click on shared. It's gonna take a second. But if you scroll down to Cyber's, make this a little bit larger, Cyber's training, click on that. And then Data Lab, click on that. And I'm sorry, we're getting there. We get to Biosciences. This is where all of the data is. So before I continue, I want to state that at any point of today, if you get lost, everything is reproducible throughout the Bioscience Data Lab Fall 24 IPYNB Jupyter Notebook. Yes. These locations are available on the wiki, yes. Yeah. I suggest for you to take this and copy it. Go back to this little folder over here on the left, click that, and right here, click paste. This will paste the notebook from the secure location onto back over here. So you can open this notebook and it has a small little, essentially all of the code that we're gonna run in today. You don't have to run much of this code, as in most of it is already been run, but feel free to follow along. So, where is it? First thing that we need to talk about is quality control tools. One of the most important and widely used tool is FastQC. Have you ever heard of FastQC? Cool. Fast quality control. And the reason why it is important is because it helps you quantify or qualify the data that you have sequenced and sent off to the sequencing people. Now, FastQC is great because it, one, tells you potential low quality scores and at certain positions. Adapter contamination, overrepresented sequences, GC content anomalies. And honestly, it's extremely fast. It does not require much. Don't get this wrong. It does not require much thinking. It's a single line to run, then you press enter and then it runs. What, however, is important to understand is being able to read what is going on in FastQC itself. Yes.
Is FastQC basically what everyone uses, or are there other options that we're just not going to today?
Honestly, FastQC is the standard. Yeah. Strongly suggest getting used to understanding FastQC. Later today, I'm going to talk very extremely little bit about MultiQC, but MultiQC is an aggregator. FastQC is the one that actually does the reports. Now, yes.
Is that you run before you align the FASTQ files or after?
Ideally, you want to run this before the alignments. And I'm going to ask you the question, why do you think that would be?
Well, the alignment process will take a while, and these are contaminated.
Right, exactly. Exactly. So literally, as soon as your data comes in, first thing you want to do is check all of your data, make sure that there is no contamination, make sure that, well, does this data make sense? And then I'm not going to talk about trimming your data today, but you might see some contamination, and therefore you might want to think, okay, maybe I have to trim some areas first before you carry on with assembly itself. Yeah, no worries. Now, the command over here that you can see, FastQC sample of FastQ-O is what is going to create the output report itself. Crap. Sorry, I have to move some windows around. Zoom is always in my way. Now there you can go ahead and run FastQC on some of the files that you want, but ideally I would like to see FastQC reports right now. So I'm just gonna go to the ones that I have already produced, and you can go there yourself as well. So once again, data shared. Okay, I'm gonna do that again. Data, iPlant, home, shared. Scrolled all the way down to Cyber's training, data lab, biosciences, and I'm sorry for all of that, but it worked out. Here you can see the FastQC MultiQC reports. Click on that, and I have created a report for one of the sequences from the reads, and open it up. This can be seen, this can be opened in Jupyter, this can be opened in your browser, but I want to run through some of the things that you see here. Now, gonna make this a little bit smaller so you can see actually what it is. Weird question. What do you see? And I'm not expecting an answer here because honestly, the first time you look at this, it's really, really confusing. On the bottom x-axis, you see the position of the read. So you can see that there is roughly a maximum of 283 reads. So you can say that there is, if you scroll up, how many reads do we have? Does it say? Roughly the sequence length is 300, between 100 and 302. And on the y-axis, it caps at 40. Thing is, it's not a great graph for understanding what FastQC is. So we have to go back to the CLI tools and look at this graph right here. So FastQC essentially breaks down the reads into positions and a Fred score, and that is what is on your y-axis. Without going into deep, these are bar plots with you have your little mustache, which are your 10 and 90th percentile, and then your little bar graph, which is your 75-25. The red line is your mean, and then you have the blue line, which is the average quality throughout. Without trying too hard, green is good, yellow is not so good, red is bad. But what are these colors? These are actually what we call the Phred score. And the Phred score is something that I very quickly mentioned last time around. And it caps at 40 because of math reasons. Ideally speaking, 30 to 40 is good. 20 to 30 is all right, and anything below that is not so great. Phred quality scores are used all the way throughout bioinformatics to understand whether a specific base is truly that specific base. For example, if this is an A, how sure are you that this is an A? The closer it is to a Phred score of 40, the better. The lower, the not so great. So when we go back to this specific run, all of the bases are absolutely great. And in this specific case, it's quite rare that an output graph is this beautiful. Something that you would see is something that I have put here, where you would have roughly 50% of your reads being good, 25% being all right, and the rest being something that you have to trim. So this is extremely rare, what you're seeing right here. But the FastQC score tells you a little bit more. It gives you this really blue graph per tile sequence quality. So essentially what this is if you do run your little Illumina sequencer, each little square inside the Illumina sequence is given a score on a heat color going from blue to red, blue being great, red being not so good. So this and this go hand in hand, where you see the small little line at the top being great, and when you see a lot of blue, you have a great sequence and you can go ahead and move on with your day. It also gives you the quality score per sequence. So here the average score seems to be roughly 39, which is a really high Phred score. And the only thing that I do want to point out for this specific report is, well, seems like most of the positions in the reader are at the beginning, which is not necessarily bad, because when you do the alignment, this kind of cancels out. Ideally, you would want to see these lines going all the way throughout, like all jogging throughout this specific graph. So this is just the only piece of information that I'm like, I'm not really sure if this is great. Oh, this is also the other one which you should know about, which is the GC content. What is the GC content? It's literally in the name.
Abundance of the Jesus.
Exactly. And what do you want the number to be? It shows in the graph. It should be a normal line that goes roughly between 49 and 51%. When you see this specific over here, GC content first peak, you know that there is something that looks weird. So that's something just to keep in mind. There is something that didn't really go too well in your sequencing. Yeah.
Oh, it's a percentage. Sorry. Okay.
Yeah. Okay.
Sorry, just a question about the last one, the per base sequence content.
Yes.
So is that saying that? Sorry, can you explain that again?
Sure. I'm going to try my best.
Okay.
So ideally, what you will want to see is a better distributed content for each of the bases. You have each of the bases on the top right, and you would want, and what you can see over here is that the best distribution is at the beginning of the read. So what's going on necessarily in the other side of the read? This is not bad, but it does raise a question, you know. But then there is another thing that is important to understand, is that these are Illumina sequences, which are really short. So always keep that in mind. These are essentially the more important ones that I need you to know about FastQC. And in my notes, I also add some extra information on each of the reports. But because of time, I have to skip up to actually showing you the report for MultiQC. MultiQC, as I mentioned earlier, is an aggregator, and I have ran here two different FastQC scores. MultiQC, I tell it, hey, MultiQC, take both of these scores and tell me more. So if you do open this file, it's the MultiQC report.html file, and click on this top left little button that says Trust HTML. Yay, graphs. I like graphs, and they are HTML graphs that you can go on and they tell you a little bit more about itself. I'm not going to stay here long because these are going through all of the data that we just talked about in represented in a different way. But what is important for you to know is that as an aggregator, it can also take data from outside FastQC. It can take alignment and the mapping data from aligners such as star, HiSet2. It can take variant calling data from BFC tools, which is SAMtools, Salmon, quantification, and differential expression. So what is MultiQC good for? It's good for reporting when you write a paper. When you write a paper and you've done your alignments and everything, and you've done your statistics, and you really have 10 different places for you to run statistics, you want to have it all in one place. That place is MultiQC. So FastQC in the beginning, MultiQC at the end. It runs exactly, by the way, like FastQC. You just type in MultiQC path to folder, and it runs. And the path to folder, the folder that holds all of the statistics. So these are the two tools. There's another one that I want to talk about, but I decided that we didn't have enough time. These are the two tools for quality control that every bioinformatician needs to know. Now, I know that all of you have been so wanting to talk and about transcriptomics, but we need to first take baby steps and understand alignments. What is the most famous alignment tool? Starts with a B, ends with a last. BLAST. So fantastic. And it's funny because BLAST is the most famous alignment tool, but this image right here, it's not a BLAST image, it's cluster-O, which is a tool which you can align things with your mouse. So if you're not happy with doing with BLAST and whatnot, cluster-O stands for cluster omega, is a great way of getting started with alignments. It just uses a lot of memory, so keep that in mind. Now, in this next section, I'm going to talk about aligners, and we're going to start with BLAST, and then we're going to walk through time very quickly because we won't have enough time, and go through BWA, Minimap, Bowtie2, Stars, HiSet2, a little bit about SAMtools, and then I'm going to have some honorable mentions of string type, Bullwagon, and DSAC2. There's a thing in the chat. Super subjective question here. What are your general thresholds for excluding a sample, Michele? 5. Literally, -5. Anything below is not great. I used to do perhaps a little bit higher than that. Are you talking about the E value, Eric? Because -1, E -5 is what I use. Evals, Fred scores, or GC content. Okay. All right, I didn't want to talk too much about this, but I usually base it on eval simply because I don't really use BLAST for Fred scores. And GC content when it comes to BLAST, I don't even know that was an option. I'm sorry. But and before we go into too deep or continue the lesson, evals is a blast only score. So when you run a blast alignment, either using NCBI or through the command line, you kind of have to understand how blast works. But the standard is always 1E-5. And it really does cut a big chunk out. Now, just mentioned, you can do access blast from NCBI. They are the ones to maintain it. I do think you can find their code from thanks, Manfred NGC for quality control, eval for blast. Yes. Eric, I'm gonna continue. Apologies. Yes, you can access NCBI, sorry, blast from NCBI. It's a really powerful tool. They have a number of computers that you can run this on. And you can, the thing about Blastic is that you can run both nucleotide and proteins and then exchange them. The nucleotide blast is blast N, protein blast is blast P. And then if you want to translate it, it shows right here, you have blast X, which translates nucleotides to protein, and T blast N, which translates protein to nucleotide. Now, running out of time, so we can't go through the exercise itself. But I have, if you do want to run this alone, and I suggest you do this at your own house, at your own home, is to get used to a little bit of what the blast output, the blast query, sorry, the blast command looks like. It's right here, and we talked about it earlier, blast dash query, blast n dash query, your gene, and I've made some genes available, of which one is a LAC-Z gene, which is the gene that creates lactose. Okay. Then you select the database and you call that with a -d flag, db flag, and where the database is, and then you say where you want your output. Now the output results.out will look something like this, where if there is essentially no blast hit, it literally tells you no hits found, move on. So you try again, perhaps a gene that works. And well, it's just going to give you what a blast result looks like. I truncated to save some space here, but there is some extra information that you can find in the output itself. I've also added some popular flags that you might want to use, such as out format. Now, if you want it in a tabular format, you want to use 6. I did not want to go there and list each single format because there is 20, and no. And talked about eval, talked about, I did not talk about max target sequences, for example, which gives you only the top 10 results and the number of threads. And this is where things kind of get, what? And this is where things kind of get ugly because we're talking about threads. From here on, you need to step back from your computer and start using either the HPC or platforms such as Cypress. Why? Because we are going to start talking about a different type of alignment. So far, or talking about BLAST, these are small sequences, but what if we need to align transcript data or genetic data, or sorry, DNA data? Okay, so we jump into our little time machine and then we go back to something called BWA. Has anybody heard of BWA? Cool, Borrow-Wheeler aligner. We are now in, I think, the 90s. And a couple of mathematicians, and we're going to go back to mathematicians here. Mathematicians are the ones that run the whole bioinformatics game, not biologists. A number of mathematicians decided to sit together in a room, mainly Burroughs and Wheeler, and decided that they really liked maths and that you can do cool things with maths. They developed three different algorithms, or excuse me, they developed a number of algorithms, and later down the line, a number of biologic laboratories decided that they could use these algorithms in order to create alignments. So now we're going from whole genome shotgun alignments to, hey, we can use computers for this stuff. So the three algorithms are BWA-MEM. Remember what MEM stands for. WAALN and BWASN. Now, the only one that really is important for you to know is BWA-MEM. Why? Because even though it was written back in the 90s, this is the algorithm that we're going to see this later on. This is the algorithms that aligners like STAR or HiSet2 use. So I'm starting at the beginning because this is where history is at. It's still a very popular tool today. It's really useful for variant calling, doing some genome expression analysis, but it's, although useful, not the one that is popular, perhaps, or the ones that I would say for you that you should use. And one important thing for you to know is that each single one of these aligners going from BLAST to BWA to STAR first requires an indexing. So the first thing that you do is BWA index reference genome, and then you move on to aligning the reads. Now, I'm writing these extremely simple commands because they will run. They will align your reads onto your reference genome. And before we go ahead, I just mentioned that perhaps something that perhaps I need to refine. Each single one of these aligners not only requires an index genome, but you also need the reads themselves. So imagine it as you go to NCBI and you're working on human. You will download the human genome first. You will need to index the human genome, and then you can align all of your read data onto the genome itself. So just making sure that we're on the same page on that. Now,
So what's happening is you've got basically a file as a reference, your data.
yes,
your algorithm.
correct. There's another thing that I just mentioned very quickly, which is the tool that you use in order to sequence. There is two schools of thoughts: short read sequencing, long read sequencing. Long read sequencing gang. Short read sequencing gang? That's completely fine with me. It's a joke because throughout my time as a bioinformatician, I did use long read sequencing. And to make it made so much more sense. It turns out that it's a lot more expensive, and my laboratory just had money for it. So I was lucky. Short read sequencing is a lot more useful for example, smaller genomes, such as E. coli or other bacteria and viruses. It would make no sense to run long read sequencing on a single bacterium. Now there is a third group of people that are coming up, and that is the nanopore people. Have you heard of the nanopore people? Cool. Little object, take a little bit of sample, put it in, $1,000, and in a couple of hours you have your fully sequenced genome at 90% accuracy, which is not that great. So it's great for medium-sized genomes. It's also a tool and a little bit of a toy, but it's a great piece of technology. So anyway, going back, BWA, great with short read sequences. Full stop. I'm just going to end it there. For longer reads, the same group that did come up with BWA created something called Minimap2. Minimap2, very useful for nanopore and PacBio. The difference comes down to the algorithm, which takes BWA and, sorry, it does not use, no, it does not use BWA and completely different algorithm for matching long reads. However, because it is the same people, they try to keep the syntax the same. Great, thank you. Now, let's jump a little bit forward in time and to probably the aligner that I worked with the most. I hated Bowtie 2, full stop. I did not understand how it worked, and I was a bachelor when I first was introduced to it. It just made no sense. There is too many words, and honestly, I didn't really like it. But it is important for you to know that it exists. Why? Because it's popular and because, well, it's still being used to the day. It first came out, I think, in the late 2000s. And once again, it uses a BWA similar, an algorithm similar to what BWA was using, implementing similar approaches to the alignment used with Minimap. So it's taking both Minimap and BWA and bringing them together, essentially. Now, one thing that I need to mention about Bowtie 2 is that it helps, or this is the way that I liked to use it. It helps with pair-end reads. Now, we are adding an extra layer of complexity here. For the geneticists in the room or online, what are pair-end reads? Yes, correct. So now we're taking into consideration pair-end reads, and ideally, they create a better alignment if you have paired-end, or a more accurate alignment if you have reads coming from both sides. So for each single alignment, sorry, each single sequence that comes out of the sequencer, you have two coming along, but it is extremely rare that you only have read one and read two. You usually have read one A, read one B, and there is a syntax that you have to follow. And all in all, this is the syntax that I am posting right here. Each single one has a comma attached to it. There is a question, I believe. How many nucleotides in a read that is considered a long read on average, and how many nucleotides on average is a read considered a short read? I will say anything between 100 to 300 is a short read, or actually, let me put it a different way. Anything shorter than 300, I consider a short read. I wouldn't really consider anything longer than 300 to be a long read. When I used to work with long reads, it will start around 1,000 base pairs. Yes, so anything between 300 and 1,000, usually what we will do is that we will sit down and say, okay, what is the best aligner for all of these reads that are neither short or long? The quick answer is using more aligners, because you have a reference genome, and then you do alignments of the long reads, and then you do alignments of the short reads. And ideally, that improves the accuracy and the quality of your overall genome assembly, depending on your sequencing. All right. So now we jump a little bit ahead of time too, where we talked about STAR. And we add yet another level of complexity, and that is spliced transcripts. Now, the reason why I hate transcriptomics is because a lot of things happen that I don't really like, such as splicing. And if you are familiar or not familiar with splicing, is that it's a process that removes the introns from a, do you know about splicing? Okay, it's a process that removes the introns from a specific transcript, and therefore, adds a layer of complexity. Now, not every single splicing is going to splice exactly at the same site, right? That is called alternative splicing, correct? Yes. So now this extra level of complexity needs to be taken into consideration. We want to do transcriptomics. What are we going to do in order to address alternative splicing? Well, thank you for stopping by. Have a great time. What are we going to do with alternative splicing? A really smart person called Alexander Dobbin up in Cold Springs Harbor decided to create an aligner called STAR. STAR is fantastic. However, STAR is extremely sluggish, as in you need a lot of power for STAR to run. As you can see, it's extremely basic syntax. So just to run the basic alignment, so without choosing additional options, requires for you to understand how many threads you have, where the genome directory is, and then specify the genome within the genome directory. And then there's this thing called SJDB-GTF file. What the hell is an SJDB? Well, Alexander Dobbin decided that he's sorry, because SJDB stands for Splice Junction Database, in the specific case, overhang. And ideally, it takes into consideration the exon to exon area. So if you know, this is me trying to explain something extremely difficult. If you know that you have two exons and an introns in between, but don't know how many bases has been spliced, you need to identify an area between these two that will then bring the exons together. Now, read length -1 is an acceptable value to use. So if you know that your read length is of 100, you want to do 99, which is, I don't really understand it myself, but just know that this helps with understanding, overlapping the two exons together and making sure that it takes into consideration the splicing, whether it's an alternative splicing or a full-on splicing. Kind of difficult to understand. He is sorry. It's 353. On to the next. And we have HiSat2. And HiSat2, unlike STAR, is faster. Why? It takes BWA aligner, and then it uses a compression algorithm. And at this point, I was like, whoa, that is so smart, because it's taking the difficulty of running STAR or the same algorithms of STAR, and it's compressing them, which can be it can be bad, because you may lose some accuracy, but at the same time is a lot quicker. So HiSat2 is good, is great. It's a lot faster, and the accuracy is all right, honestly. So we're walking through time. We went through BWA, we went to Bowtie 2, we talked about STAR, HiSat2, and I do need to point out one thing. Everything so far outputs in a single format. That is the SAM format. Anybody knows about SAM here? Cool. Fantastic. I had to blaze through the last section because I know this group is not too interested about alignments for genomes. So for differential expression analysis and transcriptome analysis, folks, use STAR, use HiSat2. So I needed to put that out there. But everything outputs into this format called SAM, and SAM stands for Sequence Alignment Map. So it's essentially a single file which has all of your reads aligned to the reference genome. It is extremely important for every bioinformatician to understand what SAMtools is and a little bit how to use it. First things first, SAMtools, or the SAM format, stands for Sequence Alignment Map. SAM is human readable. SAM is larger in file size. You want to use BAM. You want to take all of the SAM outputs and transform them into binary alignment maps. What does this do? It takes them from just being human readable to being computer readable. You don't need to know how to read that stuff. The computer does, because the computer is going to do whatever the heck it needs to do after. So SAMtools takes the output of all of the aligners, and it allows you to manipulate these sequences in order for you to have a successful downstream analysis. First thing you want to do is to take SAM, convert it to BAM. What does it do? It saves space, nothing much else. Second thing to do, and for the 15th time today, you want to index the sorted reads. Sorry, I skipped a step, dang. First thing you do, second thing you do, you want to sort the BAM file. And what does sorting does? Well, imagine that you have reads that are aligned. They don't know which genetic coordinates they're in. What SAM does is that it uses the reads around it to give it the right genetic direction. So you want to create a sorted output, which you can then index. Here you can run some statistics using FlagStat, but I need you to understand that the first thing you do, the second important thing you do is to sort things out. Then you can do this thing called an M pilot using a reference genome. And what does this do? Well, this creates a BCF file, which is useful for variant calling down the line. Now, I've introduced the idea of SAMtools. I've introduced the idea of alignments. Next week, we're going to play with those a little bit. I'm not going to be talking about every single aligner. I'm going to be picking and choosing between one of your transcriptomics alignment, and together I'm using a Jupyter notebook. We're going to walk through what creating an alignment for transcriptomic analysis looks like, and preparing it for and running it through programs like STAR or HiSet2, SAMtools, excuse me, running out, StringTie, BallGown, and these are going to be the 4 tools that we're going to be looking at next week. Why is that important? Well, if you want to do transcriptomic analysis and later down the line, this differential expression analysis, these are the tools that you require. I just had to take you down memory lane. Thank you. I won't be doing my honorable downstream, but my honorable downstream tool mention, but they are in the notes themselves. So with that said, I would like for you all to please also do a quick survey. I'm going to stop sharing over here and go and fish it. So please, if you do have time, it takes a minute. It is now in the chat, so please do fill in the survey. It gives me an idea of tools that you want to see, gives me ideas of pipelines that you want to run. and information that you might want to hear about. So thank you very much for your attention. It's always a pleasure. This is a field for me that has been close to my heart, and I truly, truly wish I knew all of this 5, 6, 7 years ago. So thank you for your patience and your attention. Have a great day, everybody.